Welcome to my channel. I'm thrilled to have you here. Today we are diving into an interesting topic, antigen. Whether you're a student, a professional, or just curious about immunology, you are in the right place. My goal is to make this topic clear and engaging, so I'll be sharing this information in a straightforward format. This topic gonna be interesting as I'll be talking about antigen in a in an introductory way. So let's get started on this first journey of our lecture, which is antigen. Thanks for joining me, and don't forget to subscribe if you want to keep learning with me. Thank you. Now, let's take a look at antigen. What is antigen? Antigen is any substance that trigger an immune response in the body. It's usually a protein or a molecule found on the surface of pathogens like bacteria, viruses, or other foreign substances. Molecules that stimulate the production of specific antibiotics are called antigens. Most antigens are large molecules like proteins and polysaccharides. Antigens are also non-harmful substances located within the body that when exposed to the immune system can fool the system into thinking that they are foreign. So what am I trying to say here? I'm trying to say that some antigens are non-harmful substances. Why some antigens are harmful substances? So they're not is not always in all cases where you can say an antigen is harmful. So there are times where antigens are not harmful substances located within the body. In this way, the immune system can be fooled and will think that the antigen is harmful. Now let's take a look at the location of antigen in the body. There are three primary places where you can see the antigen in the body, which are in the blood, the lymph, and the connective tissues. Now let's take a look at the location of antigen in the blood. So monocytes travel in the bloodstream and this monocyte act as antigen presenting cells. The second location is the Kupffer cells in the liver and the phagocytic cells, which are located in the wall of blood vessels in the spleen, remove foreign antigens and inactivate them within their cytoplasm. So what am I trying to say here? I'm trying to say that the Kupffer cells and the phagocytic cells, they remove foreign antigen and inactivates them within their cytoplasm. Now look at the connective tissue. So at the connective tissue, we have the loose connective tissue, which include collection of unencapsulated B and T cells called diffuse lymphatics that are located in the lamina propria and pars papillaris, hinders antigen from entering tissue. What am I trying to say? I'm trying to say that in the connective tissue, we have loose connective tissue. The loose connective tissue, we have the B and T cells that are located in the lamina propria and pars papillaris, which hinders antigen from entering into the tissue. Now, the location of the antigen in the lymph. Let's take a look at the lymph. Antigen in the blood will circulate through the lymph system and carried into the lymph nodules and destroyed by waiting B and T cells. What am I trying to say? I'm trying to say that in the lymph, there are antigen in the blood and they are circulated through the lymph system. And these antigen are carried into the lymph nodules and they are destroyed by waiting B and T cells. Remember, I said earlier on that in the connective tissue, the B and T cells are located in the lamina propria and the pars papillaris that hinders antigen from entering the tissue. So in the lymph nodules, the the antigen is carried into the antigen is carried through the lymph system into the lymph nodules and then destroyed by these B and T cells that are located in the lamina propria and the pars papillaris. So let's take a look at types of antigens. We have different types of antigen ranging from exogenous antigen, endogenous antigen, the autoantigen, the tumor antigen, the allergens. Now, exogenous antigen, where is this exogenous antigen located? They are they are coming from outside. So they enter from outside the body like bacteria, virus, pollens, or allergens that come into the body to destroy the tissues. So this type of antigen is called exogenous antigen. So we have endogenous antigens. The endogenous antigen are generated within the cells in a scenario where there is infection occurring inside the body or where there is abnormal cell processes like in cancer. That's this type of antigen generated within the cells is called endogenous antigen. They are in form of infection or in form of cancer occurring in the body. Now let's take a look at the autoantigen. These involve parts of the body own cells that they are self antigen. So they may just have they occur in the body. They are self antigens. They are the one they occur in the body. The body might just generates it and think that it is harmful and they harm the tissues. So we have the tumor antigens. They are found on cells, they are found on cancer cell and often unique to tumor. So they are tumor antigens. We have the allergen. So this is typically harmless substances like pollen or certain food that triggers an immune response in an allergic person. So let's take a look at the application of antigen. Antigen is applied in the world of vaccine, in the world of blood, and in the world of cancer immunotherapy. So let's take a look at antigen application in vaccines. In vaccines, introduce a harmless form of antigen to train the immune system without causing illness. So in the vaccine, we can use antigen in the world of medication to train the immune system without causing any illness. So in the world of blood, we have during transfusion, this antigen plays a role. It plays a role on the red blood cells, and matching this antigen is essential for safe transfusion. So in the world of cancer immunotherapy, targeting tumor antigen allows the immune system to recognize and attack cancer cells. So through application vaccine, the blood typing and the transfusion and the cancer immunotherapy. So guys, this is just a brief introduction of the antigen. On our next topic, we're going to be talking about the antibiotics. These antibiotics will state the connection between antibiotics and the antigen in a lecture format. So I think I will be doing, in our next lecture, I will be doing paper on board, so you all will understand what I'm trying to say about the antigen and the antibiotics in a more detailed way. Thank you. See you guys in our next lecture.